Listeria monocytogenes is a bacterial species of concern, especially in the food industry. It's not the leading cause of foodborne diseases, but it is one of the leading causes of foodborne deaths. And it's great concern because of that reason for the FDA, USDA, and other organizations. It causes an infection called listeriosis, which results from eating contaminated foods, and it really affects pregnant women, elderly, immunocompromised people, essentially people of great danger. And so now we're going to dive into exactly why it's the case and what's the problem. Hello, everybody. I'm your host, Dr. Yu. This is our channel. This is Microbiology Explained. We're back to our food safety series. Today's video is going to be about Listeria monocytogenes. In this series, we're going to dive into the ins and outs of food microbiology. We're going to talk about the good actors and the bad actors with respect to food. This is the first episode in a series of videos that we're going to make about organisms per se, the organisms that are of interest to us in the food industry, and of you who are a layperson who may be interested in what particular foodborne diseases are out there and what is of concern. So, Listeria monocytogenes, it's a bacteria that's ubiquitous in the environment. It can be found everywhere: soil, water, effluents. It can be found in a variety of foods, which concerns us. It can be found in the feces of humans and animals. It can be found in a diverse range of environments. The organisms its discovery dates all the way back to 1924 by three gentlemen, Murray, Webb, and Swan. They isolated Listeria monocytogenes as the causative agent for a systemic disease infecting rabbits and guinea pigs in their lab in Cambridge, England. And since then, it's been studied extensively. Why it's of such great interest is because its biology is hardy. It's a hardy, salt-tolerant organism. But unlike most pathogens, it can grow at really cold temperatures and survive. We're talking about 1 degree Celsius or about 32 degrees, 34, 35 degrees Fahrenheit, very close to freezing temperatures. Why this is of concern is because most places we use the cold to control pathogens. And here's the primary method of control of pathogens does not affect this organism. Of the members of the genus Listeria, there are many members. In some papers, I have read up to 11 or 22 members. It includes many species. Of those species, monocytogenes is the only one which has consistently been associated with human pathogenic illness, and L. ivanovi has been associated with pathogens and ruminants. The pathogenicity of Listeria is interesting because it doesn't affect you immediately. The clinical course of the infection usually begins about 20 hours after ingesting of heavily contaminated foods and causes cases of gastroenteritis. However, the incubation period for the illness generally can be much longer than that. It can be up to 20 or 30 days. So it may be a while after you've consumed it before you actually begin to show symptoms. Like I mentioned before, the population which should be most concerned is pregnant women, elderly, immunocompromised patients, and the like. There are two main types of listeriosis: non-invasive listeriosis, which is a mild form of the disease, which mainly affects healthy people. The incubation period for this is a short day. And then severe listeriosis, which is a much more severe form of the disease and affects the high-risk groups that I mentioned before. This incubation period can be up to 1 to 2 weeks and can vary quite a bit. And so it can be a while after consumption of infected foods that one can actually show the symptoms. Symptoms usually occur with muscle aches, nausea, vomiting, some diarrhea. In severe forms, the infection can spread to the nervous system and can cause stiff neck, confusion, and convulsions, even something as serious as that. The infectious dose has not been clearly defined or determined across the board, but in some cases, like in raw and inadequately pasteurized milk, as few as 1,000 Listeria cells can cause the disease. And unfortunately, the mean mortality rate in human beings can be as high as 20 or 30% if the right treatment is not applied in time. To detect Listeria in a clinical setting or in a food industry setting, most of the times you go through an enrichment broth, and then from that enrichment broth, the identification can occur. However, nowadays with new molecular techniques and kits that can be PCR-based or ELISA-based, much more rapid analysis. We're talking about 2-3 day turnaround time is possible, at least to identify it down to the genus, if not to the particular species. In another video, I will delve into the details as to such techniques and compare the pros and cons of those techniques. Food that it's associated with are, as I said before, pasteurized milk or under-pasteurized milk, chocolate milk, some ice cream, raw meats, poultry, fermented sausages, hot dogs, deli meats, raw oysters, raw fish. So a lot of wide range of foods have been associated with Listeria, including raw vegetables. One of the most notorious outbreaks of Listeria back in 1998 and 1999 was a large-scale multi-state outbreak which resulted in 50 cases across 11 states, and 6 adults had passed away from Listeria infection, and it was linked to contaminated hot dogs. And that was the case there. The biggest thing to do to prevent Listeria is safe handling of food. Like I mentioned before, the preventative measure that we use for most foods is chilling and refrigeration, and that does not work as adequately for Listeria. So it's important for you to have good manufacturing practices if you're in a manufacturing environment, or good handling practices if you're in a kitchen or a restaurant or something like that. That involves cooking food thoroughly, separating cooked and raw ingredients, safe water and raw materials, proper hand washing techniques, and things of that nature. The cure, it can be treated through antibiotic treatments, generally ampicillin, penicillin, and other antibiotics of that class. However, the treatment has to be timely in order to have the best impact. So in summary, Listeria monocytogenes is very common. It is present in the environment a lot of the times. Listeria genus as a whole is present in the environment. Listeria monocytogenes is also present, like other members of the species. And it's highly resistant to some preservative methods like salt or smoking, and it's able to survive in the low environments, low temperature environments, which is why it's a big concern. But the best way to handle it and contain the species is through adequate, good manufacturing practices, good handling practices, and a rigorous program, in the environmental program, which goes out to seek to detect Listeria in any harborage points that may be. Once again, thank you for joining us in this video. Thank you for watching. Like, comment, and subscribe. Let me know what you want to know more about, or parts of food microbiology interest you the most, and I will see you all in the next video. Thank you very much.